Hello everyone, today we're going to be talking about carbohydrates and tripeptides. Alright, so what I've drawn here is a form of a carbohydrate. Okay, it's actually a disaccharide. Now we'll get to what disaccharides and monosaccharides actually are and what that means. But just for the moment, if we just recognize that di means two, and saccharide actually, you can think of it as meaning carbohydrate. So two carbohydrates. Alright, so that's what a disaccharide is. In this example here, we have two glucose molecules coming together to form a disaccharide called maltose. And then also as a byproduct, it produces water as well. So to start off with, let's talk a little bit about what glucose actually is. So glucose can exist in two forms. It can exist like this here. So this molecule here is one glucose monomer, alright, which means it's a simplest form, the simplest unit that a carbohydrate can be is a monomer. So if we just took half of this molecule here, just looked at this left side, then that would be a monosaccharide, because it's just one glucose molecule. But then if we put them together, we form a disaccharide, which is two glucose molecules put together. Okay, and the diagram for this, so the actual formula for glucose is in the formula book as well. So you'll see in the formula book that you'll have a few different forms of the same thing. You'll have glucose in this form, in the cyclic form, which is called alpha D glucose when it's like this, and you also have straight chain glucose too. There's a few things I want to talk about with glucose, a few things to recognize. I'm going to draw the straight chain here as well, so we can have a look at an individual glucose unit. Okay, so now I just want to focus on this molecule here. Okay, so this is a glucose. It's also a monosaccharide because it's one unit of glucose. So this is actually equal to, just put down here, this is glucose. Alright, but when it's drawn like this, it's actually straight chain glucose. So it's not in a cyclic form, which is what we have here, but instead it's in its straight chain. So if we just look at this one glucose unit, a few things to note. There's usually about 5 or 6 carbons in the carbon chain, or in the carbon backbone. So if we count the carbons for this glucose, we have 1, 2, 3, 4, 5, 6. So there's 6 carbons. Alright, and we also have a double bond to oxygen on the first carbon as well. So this carbon here, although it's not drawn in your formula book, it actually has a double bond to this oxygen. So because there's a double bonded oxygen on that last carbon, it actually means it's an aldehyde functional group. So a few things to note as well. We have lots of these hydroxy groups, these OH groups, scattered throughout the molecule. So you have that alcohol functional group too, and we also have the aldehyde functional group on that first carbon. So it's a bit strange because it's kind of a combination of the two, but you don't have to recognize, you don't have to name this in terms of the IUPAC naming. Instead, you just have to recognize that this is glucose. Alright? Some things you might be asked to do in your exam is to describe the features of glucose. So you can say that it has 5 carbons in the carbon chain, alcohol functional groups, and also that aldehyde functional group. Now, there's also another kind of simple sugar that's drawn in your formula book, which is another monosaccharide, and that's fructose. So just underneath that glucose drawing in your formula book, there's another one that has fructose. That is nearly identical. The only change is the double bond to this oxygen molecule on the first carbon is somewhere in the middle of the chain. So we have a double bonded oxygen to the carbon on one of these carbons here. So that's the main difference between the straight chain version of glucose and fructose. You can see the position of that double bonded oxygen in different places on the carbon chain. Okay, so that's pretty much the main features of a glucose molecule, and a fructose molecule is very similar. remember the only thing that's changing is the position of a double bonded oxygen to the carbon. You'll see that in your formula book as well. Okay, so now let's move back to the side here. So this molecule here, this straight chain, is exactly the same thing as this molecule here. All right, so these two are the same, just drawn in different forms. Okay, so it can exist in both the straight chain and also the cyclical form here. All right, this diagram here, I've actually drawn two glucose molecules side by side. So you'll notice that there is a bond in the middle. So this arrow here is indicating the glycosidic bond, and that is basically the oxygen bridge that is holding these two glucose molecules together. Okay, so these two glucose molecules coming together and form a glycosidic bond, and then they produce a thing called maltose, all right, and also water. So when these two glucose molecules come together, they will form water as a byproduct, as well as the main disaccharide that they produce. So main things to note with carbohydrates is they can exist in both straight chain form, cyclical form, and they are formed, so disaccharides means two carbohydrates, monosaccharides means one carbohydrate, monosaccharide, disaccharide, okay, and disaccharides are formed via a condensation reaction, okay. So they're the main things to recognize with carbohydrates. Right, one more thing I want to mention actually with these carbohydrates is you'll see it drawn three different ways in the formula book. So in the formula book, you have this one, and then you'll have another one next to it that looks a little bit like that one there, but it's slightly different. So the first, on the left side of the formula book, you'll have this molecule, and then the next molecule drawn is actually the same thing as this, just drawn in a different way. Okay, this is the same thing, but drawn as a kind of a straight line drawing, not showing all the different bonds. So you have this, and then that other form, and then after that, you have this one here, which is the cyclical form of glucose. All right, it's important to note that those first two diagrams, so the left side one and the middle one, are exactly the same thing, and then the one on the far right is glucose in its cyclical form. Okay, so a little bit different that last one on the right, but they're all variations of the same thing, of glucose. Same with fructose as well. All right, now let's have a look at tripeptides. All right, so I want to have a look at this molecule here a little bit. So what I've drawn here is a tripeptide. The reason it's tri because we have three. So tri means three. Peptides means you can think of as peptide bonds. And peptide bonds only occur between amino acids. So tripeptides is three amino acids that have come together to form the structure here. So the three amino acids that have come together for this example is glycine, serine, and alanine. Now, if you have your formula book with you, it would be really helpful to kind of find them on your formula book and try to match them up with the different parts of this tripeptide here. So to draw these, most of it is just copying from your formula book and then putting them together via the peptide bond. So what is a peptide bond? Well, this part here, so the link between all of these amino acids is a peptide bond. So it's actually this bond here. So that there is the peptide bond. Okay, and we have another one here. So peptide bond, think of it as the bridge between the amino acids. So just like we had a bridge between the two glucose monomers in the previous example, this is a bond between two amino acids. Okay. So peptide bond 1, peptide bond 2, and then we have our amino side of glycine on the left, so our amine side, and then on the right here we have our acid side of alanine. So a few things to note with these amino acids is they're very similar. So each one of these is very similar. It's basically just repeating itself over and over again, this top part here. But the thing that's different is this side chain. So I'm going to get a different color here, and we can actually see a different side chain for these different molecules. So look at this here. This is the side chain for glycine. This here is the side chain for serine. And then this one here for alanine is different again. So that's sometimes referred to as the side chain or the R group for amino acids. And it's basically what gives you the different types of amino acids. So depending on what's in this chain here is what gives you a different kind of amino acid. And you'll see that in your formula book as well. If you go to your formula book and you have a look at all those different amino acids, this part here stays the same. The thing that changes is these side chains. Okay? And because these top parts stay the same, it's very easy to draw tripeptides. So you're basically drawing this part over and over again with different side chains. So if we look at a amino acid by itself, we'll have a NH2 end on this side, and then we'll have a acid end on this side. So I'll draw a amino acid by itself so we can have a look. Alright, so what I've just drawn here is glycine when it exists by itself. So this part here and this part here is exactly the same. It's the same molecule. The only part that's different is we don't have this OH group anymore attached to the end here. So these amino acids are actually formed in condensation reactions. So when they come together, there's going to be the OH group from glycine and also the H group from serine come together and they form a water molecule. So I'll draw serine so we can have a bit of a more idea of what's going on. Alright, so now we've got glycine and serine here. Alright, so this peptide bond comes from this part of the two molecules. So it's formed between the OH group of glycine and the NH2 group of serine. So when these two ends come together, they form water. So we know it's a condensation reaction. So I'm going to put here the condensation. So H2O is produced. Okay. So if we know that water is produced, we know that the two hydrogens and the oxygen have to come from this molecule here. Alright, so we're going to get the oxygen and one of the hydrogens from the OH group of glycine. So this OH group is going to come off the molecule, and we're also going to get one hydrogen from the NH2. So that's why, if we look up here, we're only left with NH, and we don't have a OH group anymore, and we lose one hydrogen from the NH2 group. So that's why the peptide bond doesn't have OH or one of these hydrogens involved. So it's only the NH molecule that is left on the serine amino acid. Okay, so that's the peptide bond, and that's how that occurs. And that'll happen up over here as well. So the same thing will happen with serine and alanine on the next amino acid. Okay, that's everything that we're going to cover with tripeptides. If you have any questions, let me know. And thanks for watching.